Hi, my name is Piyumika Suriyampola. I'm a postdoc at Arizona State University. Today, I want to share some results from our study that investigated how co-occurring environmental perturbations affect sensory motor behavior. With the rapid growth of human population, many habitats experience drastic changes to the visual environment, such as artificial light at night, increased turbidity, and eutrophication. And these changes have the capability to affect organisms at multiple levels. For example, extended exposure to artificial night light affects circadian rhythms of isopods. Low visibility resulted from eutrophic conditions are known to alter mate choice in cichlid fish, and the poor visual conditions created by turbidity trigger zebrafish to shift their reliance on sensory cues. While individual impacts of altered visual environments on organisms could be significant, these habitats often experience multiple perturbations at the same time. The way visual environment interact with these different perturbations may result in more complex alterations in organisms. For example, when hermit crabs were exposed to boat light along with boat noise, they tended to be more distracted from predators than when they were exposed only to boat noise. In our study, we focused on multiple interacting environmental stresses and examined how the exposure to poor visual environments, like the conditions created by turbidity or eutrophication, along with plastic pollution, affects visual motor behavior. We specifically ask whether the combination of low light and pollutant exposure has a greater impact on visual behavior than being exposed to either of them separately. We already know that prolonged or developmental exposure to poor visual conditions result in visual impairments. Here we focused on small change in visual environment and then combined it with environmentally safe levels of bisphenol A to investigate how this interaction affects visual response. Bisphenol A, which is commonly known as BPA, is widespread in aquatic habitats because of its role in synthesizing polycarbonate and plastic products. It is also an endocrine disruptor, which is linked to a myriad of outcomes ranging from diabetes in humans to decreased reproductive viability in fish and learning and memory impairments in rodents. Despite the demonstrated effects of BPA on the central nervous system, we know little about its influences on the peripheral nervous system, specifically on the visual sensory system. Here, we maintain zebrafish embryos either in low light or regular lighting conditions until 7 days post-fertilization. We exposed half of the individuals in each lighting condition either to water control or environmentally relevant concentration of BPA. We tested their locomotor activity in open field test, and using optomotor assay, we determined their response to a moving visual cue. When we examined their response to a moving visual cue, we found that 32% of zebrafish that were in the control group responded to this cue. And there was a slight decline in response when they were exposed either to low light or BPA. But this decline was not statistically significant. However, when we combined these two treatments, it resulted in a significant decline in optomotor response compared to the control group as well as to the individual treatment groups. This indicates that the combination of low light and BPA is sufficient to create a significant deficit in optomotor response. Since movement of an individual is required to display optomotor response, we then examined whether there are any differences in locomotor activity in zebrafish exposed to different treatments. Even there were differences in the distance zebrafish traveled in the control, low light, and BPA treatments. These differences were not statistically significant. Interestingly, when we combined low light with BPA, the distance zebrafish traveled still did not differ significantly from other treatments or the control group, which indicates that the change in visual motor response is triggered by a change in sensory mechanisms. Overall, we see that being exposed only to low light conditions or environmentally relevant concentrations of BPA is not sufficient to create a deficit in visual motor behavior. But when these perturbations occur together, they result in the highest deterioration of optomotor response. With no change in locomotor activity, we can suggest that the interactive effects of low light and BPA are acting on sensory mechanisms to alter visual motor behavior. These findings provide alarming information on how environmental stresses can alter behavior even at low levels when they co-occur with stresses that complement its functionality. Thanks so much for watching this video.